Yeah, so my obsession with structure and function will be on full display once again in my presentation here. And I think it's, on the one hand, a very central notion in biology, not just molecular biology, but especially there with all of its reductionistic tendencies, and thus also being fitting, I think, for the spirit of the symposium, not just for the reductionist part of it, also with the famous what is life question that at least became very famous with Schrödinger's little book, What is Life? So lots to find and unpack in molecular biology, where I also have a double background. Besides being a philosopher, I spent my fair share doing wet lab experiments as a molecular biologist as well. And the structure function notion has never ceased to fascinate me. And I still think it's a very central problem that I want to talk about today. It's also fitting to title this whole thing about two dogmas of molecular biology, since the molecular biologists themselves are not shying away from using this term of dogma, because they have the famous central dogma of molecular biology due to the legacy of Crick. Unfortunately, kind of, because he, as he later admitted, just introduced the term for not knowing what dogma actually means. And having already used the term hypothesis, that would have been more fitting in another context of the sequence hypothesis. So there we have it. And there's still this flavor of something that a true believer cannot doubt with the central dogma, and it's found its ways into all kinds of textbooks. And this is again quite unfortunate, because the schema that's presented there with the label of central dogma, first of all, is not the thing Crick had in mind, which was just only about the flow of information, a very specific hypothesis. But now students and the mainstream molecular biology is stuck on this kind of scheme, but there is this kind of linear chain of determination, also with this short slogan, often sometimes DNA makes RNA makes protein, that there would be, in also again a very reductionistic fashion, a determination relation chain where you start out at the level of DNA with a nucleotide sequence that would determine the RNA sequence, which then in turn would be determining the sequence of amino acids in the protein, and then going on even to claim that then the sequence of amino acids would determine the structure of a protein. Then this structure, again the notion of structure, which also was nicely featured in the previous talk, and we'll have more to follow up on in the Q&A, like in the discussion forum later. The notion of structure here being famously linked to the notion of function, and then basically all biology, all the roles, all the magic would just happen if we follow this chain of determination relations being traceable down to the sequence of nucleotides in the DNA. This is the kind of textbook picture you will get in most biochemistry textbooks. So the two specific dogmas I want to pick out here and want to talk about today is this first one, that there is this determination relation between a sequence and the structure of some macromolecules at least, and then that structure would be determining function. These are the kind of two dogmas I want to tackle and unpack a bit. In good analytical tradition, it will be about asking questions about what the terms in these statements mean. First of all, what is meant by the term structure? What do people mean when they say function? What kind of relation is being expressed with this determination predicate? What kind of relation can be meaningfully drawn between sequence, structure, and function? And then also think about what this role, what kind of roles this relation could play in terms of explanation, understanding, in reductionist terms and whatever. So this is certainly a very rich relation that's hiding behind this notion of determination there. So these are also the respective questions I'll split my talk into. If you look into a keyword search, you could of course use any kind of keyword these days with the exponential growth in publications, and you'll find similar graphs or all kinds of keywords, but certainly there's something unique about structure and function. So 2023 is not over yet, so I'm pretty sure we'll have a trend continue and have a higher peak there once the year is over. And 2024 is already starting to grow too. But maybe these numbers are not so reliable, but if you just walk into your standard molecular biochemistry library, you'll find all these kind of textbooks where structure and function will be prominently featured on the front of these textbooks, sometimes form and function. So clearly scientists think that this is something very central to them, but unfortunately then it often remains vaguely at this label stage or at the primitive schema I showed you at the beginning, and it's never really unpacked and looked into the rich notions that they work with there. So hopefully this is where philosophers can step in and work with these very rich notions. Well, it's been acknowledged that there is something important about the notions of structure and function, especially in molecular biology, where Richard Lewinson there claims, especially when we look into the disciplinary history as well, why is biochemistry a different thing than molecular biology? And one line of argument often goes that there is this unique mode of explanation specific to molecular biology that puts the three-dimensional structure of individual macromolecules at center stage and become the basic element in predicting and understanding. So thus molecular biology would not just simply be the biochemistry of a certain class of some interesting biologically important compounds, but rather it is and here is the interesting formulation here, a reformation of explanatory narratives in which the individual molecule and its detailed three-dimensional structure are at the center of explanation. So there we have the explicit reference to the centrality for explaining with regards to at least one kind of structure, the three-dimensional structure, but I'm not quite sure we can leave it at that. So certainly I think this is where a lot of the focus has been in the past, where structural biology has been very strong and continues to be very strong. But I think there is more to the notion of structure here than just this one three-dimensional one, and we'll come into that. So if we want to further delineate what makes molecular biology and its explanatory practices so unique and sets it apart from other areas like biochemistry, again, it's really nicely put here by Richard Burian that molecular biology would be less like Newtonian mechanics than it is like auto mechanics. I like this comparison very much. So there we would be looking into certain mechanisms on all different kinds of levels, but of course all the way down to the molecular level, and specific importance being given to the molecular level, and thinking also that somehow this molecular level would suffice to explain all the way up again. But this other direction of all the way up is often forgotten or neglected, but I think it's, at least with the intention of what these explanations are supposed to do, is also to work not just all the way down, but again backwards all the way up to also explain organismic structure, functional elements, development and evolution, where structure and function should be used for these explanatory narratives. But for the philosopher of science trained in the classical physical sciences there, the field of the life sciences have often been a bit unsatisfactory, but because we don't have such a neat Newtonian or quantum mechanics to work with. But molecular biology is a bit messy. Like it's still a unified discipline, as is being claimed here in virtue of the powers of its techniques, and especially with working with all these different levels, all the way down to the molecular, at the cellular, at the organismal, at the physiological levels. But the synthesis of problems of form or structure and function are again implied to be the central pillar of this whole enterprise. But still, what exactly is going on here? The synthesis of form and function, despite being central, is still ill understood. It is historically important feature of molecular biology, but still I think there is many question marks remaining, and hopefully we can put some into that. So let's unpack what could be meant by structure. So here's just the suggested distinction I would put forward, that we would put the suffix sec for sequence structures, like it's a one-dimensional primary structure where you just have, like here, the sequence of amino acids in a string. On the other hand, we have the three-dimensional shape that has already been mentioned, and that probably is what people think about most when they speak about structures in molecular biology. But often they also mean the primary structure, but at least these two can be neatly distinguished. But then for certain kind of explanations, it's necessary to just don't look at individual components, but really look at network structures, and systems biology has been riding this horse very rigorously, putting emphasis on these organizational structures and the mathematical models being used there, also putting dynamics and all that kind of in. So this is at least an additional notion of structure that biologists are using when they want to explain functional features of what they are working with. And we'll find different kinds of structures on different levels of organization, and the more we move to this abstract universal level, we'll be dealing more with this organizational structure kind, where it's more the topology and dynamics of these abstract interaction networks, where we are not so much interested into the molecular three-dimensional details of these individual components. On the other hand, we have the sequence structures there that are relating to the three-dimensional ones. So we have all these three notions of structure present on the kind of levels that biologists and life scientists are working on. The prime example would probably be to think about proteins. That has been what I've been working on mostly, but of course there's many other structural components, also the more abstract mathematical ones that would be interesting to look into when explaining how structure and function are being related there. So you've all probably seen this picture from Watson and Crick, where they, in the British understatement, also put that it has not escaped our notice that the specific pairing, some structural thing we've postulated, suggests a possible copying mechanism, something functional for the genetic material. So again, structure function being prominently featured there. You might not have seen this one, but it's in the same rod and stick and metal and cardboard, working with representing protein structures here. The first one by Ken Drew, and also producing paper models back in the day. Nowadays we have modern computer graphics, and you've probably all seen this from AlphaFold or whatnot, mathematical models. I'll have a few data slide. But here the good back old practical effects were still good to representing some structural features, I think. Okay, so much for the easy part. Let's open Pandora's box and talk about the notion of function, which is always a hornet's nest, especially in a group of biologists, with this big decade-old fight, I would say, between these two schools, the selected effects account of Wright and the more causal role notion of function that can be traced back to Cummins. I don't want to go too much into the details, but I think we also, as with the notion of structure, we should distinguish between different notions. And I found Arno Wouter's 2003 distinction between 4 different notions of function useful. And I'll also use 3 of those actually to make my suggested distinction that we should distinguish between function as a biochemical activity that can be attributed to an individual macromolecule, a single component. That we should also have the notion of a functional role, the biological role, something it plays in a larger mechanism that cannot be attributed just to an individual component. For example, something like upregulating blood coagulation. That's not something you can just attribute to a single component. It needs to be part of an interaction network, an organizational structure to say something about that, and that would be the biological role notion of function. But everyone's favorite, at least besides me, I'm not so obsessed with this one, but this is where a lot of the attention is going in the philosophical community, is the selected effects notion of function, why something has been selected for a specific task. Of course, nothing makes sense except in the light of evolution. It is a central notion of function, but for reconstructing a kind of determination relation that's allegedly happening from structure to function, I would find it very odd to bring the evolutionary history into this, like that the structure of something would determine its evolutionary history. That sounds so wrong to me on all kinds of levels. So there is something to be told about, again, maybe in terms of constraints, that why certain structures are selected for certain functions, or the other way around. Probably, arguably, we could say evolution is targeting functions and doesn't care about structures whatsoever. And there we can say something about why something has been selected rather than something else, but that's not something we can bring into this structure-function relationship directly. It's more a story about the evolutionary history and trajectory of this relation. With this suggested distinction, I think I'm also in line with scientific practice, where some vocabulary has been introduced for attributing functional terms to molecules. Very old enzyme commission that's just about catalytic components. That would be a biochemical activity that can be attributed to individual molecules. But the big player is, I think, the gene ontology vocabulary there. And I think that maps, on the one hand, nicely with their distinction of molecular function to my biochemical activity. Biological process, I think, captures the idea of biological role quite nicely. For evolutionary selected effect function, there is no term in gene ontology. And weirdly enough, cellular component is considered to be functional in the gene ontology vocabulary, but it really only tells you something about the location of a component, which is useful information about what it does functionally, but it's not by itself. I would actually consider this much more as structural information that can be informative about function, but it's in this vocabulary. So if you're on board with me with making this distinction between three different notions of structure and three different notions of function, we can try to put things together. And here is the suggested meaningful relations I think we can draw there. So we would have the sequence, the primary structure that is relating to the three-dimensional structure. That would be the first dogma, Crick's sequence hypothesis. There's a lot to say about the whole protein folding problem there. It's not as directly and straightforward a thing, even if with AlphaFold people now are claiming that maybe the protein problem has been solved. I don't think it has solved it at all. It's a very useful prediction tool, but that's a different story. But then we can continue to connect the three-dimensional structure to this biochemical activities and organizational structures to the biological roles, notions of function. And then we can build higher levels of organization. We could have this one structural organization being a node in another higher level structural organization, and we can attribute maybe a higher level function to that and go on about this. Whereas the selected effect evolutionary notion of function, I think, is really about some evolutionary changes in these structures and functions, rather than about a relation between structure and function by itself. That's my argument. And here is a Godfrey Smith's quote that I really like. Let no philosopher join what science has put asunder, when philosophical discussions are so often exclusively focusing on this selected effect notions of function. I think there is an interesting story more about the structure-function relationships if we are more on the causal role side of the function debate. What kind of relation do we now want to draw between these kind of pairs? I think that's an open philosophical question, in part also answerable empirically once we have the clear made distinction here. My hunch would be, at least for some of them, especially between the three-dimensional structure and the biochemical activity, some kind of supervenience relation might hold, that whenever you see a difference in the biochemical activity, you will find a difference in the three-dimensional structure. I think with the protein folding problem, not even supervenience will work because there is redundancy in all of these kind of things. But at least we can use it for predictions when we have the relations between these kind of pairs. And the real confusion, I think, is really only when things get mixed up and when people are trying to attribute to a sequence of amino acids or even of the DNA and say, oh, this sequence has this biological role. I think these are meaningless statements, really, that are jumping across levels, really category errors when functional attributions are being made to a different kind of structure. So here's a bit of a graphical abstract I try to do. I'm not sure if it is helpful at all. Let me quickly walk you through it. If you start out here at the root with the sequence structure, here we can have a relation to the three-dimensional structure of a molecule, which by itself would have some biochemical activity to it. Then we have maybe an ensemble of a couple of such individual molecules with their three-dimensional structure, which are themselves organized in a network. And this structural organization then can be attributed a biological role function, you have another network of these, and you can attribute that a higher level biological function, and so on, all the way to higher levels. And again, the selected effects notion story is on a different page here about some changes in how these relata are connected. So let's go back to this linear chain of determination I showed you at the beginning. If we start out with the nucleotide sequence determining the amino acid sequence, it's often referred to as the central dogma, even though for historic reasons I don't think that's quite accurate to do so, because Crick has been very specific again about just the flow of information with some, but again, the textbooks have made it so popular, let's deal with it. That the amino acid sequence is determining the protein structure, especially the 3D structure, is known in the literature as Anfinsen's dogma, because he observed first that some proteins would spontaneously fold back into their native conformation. There's lots of exceptions there, but again, it's with this loose, weird notion of dogma that only we find in molecular biology. And for lack of a better name, I call this last part just the structure-function dogma, that the protein structure would determine its function. But as you know by now, there is more unpacking necessary with, hopefully, these three notions of structure and function helping clarifying where we can make meaningful connections there. Let's quickly talk about some of the difficulties. First, with the Anfinsen's dogma there, people have actually taken up a kind of sports where they are creating maximum similar looking chains of amino acids. Here the sequence is almost identical with just a few changes in amino acids. For any protein structure predictions, often we say, oh, they will have to look basically the same, but as it turns out, they are folded completely differently. They have also very different structural properties, as you would have thought. And so even just thinking that a similar sequence structure would guarantee you a similar 3D structure is empirically wrong. In fact, another kind of dogma that was nicely featured in this Nature paper here, that proteins are really neatly organized in these three-dimensional structures that we've seen, is also a bit of an overstatement. Many proteins exist intrinsically unstructured, so there is no three-dimensional conformation to them. They will wiggle around, they're very dynamic, changing all the time depending on their interaction partners. So also maybe the interaction boundaries is something very alive and kicking in the world of proteins, and not so much a pre-given thing for many things. Of course, we have AlphaFold, who's made it through all the headlines recently with claims that it has solved the protein fold problem. No, it hasn't, or at least that it's such a wonderful tool that we don't now need to do the painstaking X-ray and nuclear magnetic resonance experiments anymore. We can just run it through the computer. The AI will be doing a much better job than all the physically informed models we've come up so far. And again, claiming that the structure can facilitate a mechanistic understanding of the function. So the structure determines function dogma, again, growing very strong with the expectations being brought to the table. But even if AlphaFold is very successful at predicting proteins, and it still does a very shit job at predicting transmembrane proteins or things that are simply not in the database that it has been trained on. So there's many more problems. But even if it were a perfect predictor, which it's not, how would it really inform our understanding of the function by itself? So the three-dimensional structure can be limited in telling us something about the biochemical capacities. Like if we know this fold typically binds zinc, and then it can bind as a transcription factor a DNA sequence that is look like this, we can make some inferences. Yeah, sure, but again, the big reductionist promise, we have uncovered the blueprint of life. It was first a Human Genome Project, but we couldn't even predict proteins after that. Now we can predict proteins. Haha, now we are back in the game. Well, again, the best you can do is predict three-dimensional shapes and make some very specific predictions about the functional components. Again, there is also a function prediction contest. AlphaFold dominated the structure prediction contest for 2 years in a row now, but for the function, we are not even close to anything meaningful, I think. So still long way to go to move from structure to function. But for structure prediction, it's a nice tool, this AI model, but again, I would be very cautious with the big claims being thrown around with this reductionist mindset often. So I think I'll have to agree with Sandy Mitchell and Angela Gronberg here that even with the successes of AlphaFold, that the X-ray crystallographers are still going to be in business, and also the experimentalists working at figuring out the functional side of proteins and how, let alone what physiological roles these components will play. I don't think the AI has yet some good solution to offer there that could even think about replacing the experimentalist. So there's a bunch more problems to this idea that structure determines function. On the one hand, we have the large class of intrinsically just unstructured proteins, some of which are actually essential, that certain functions are exclusively implemented by intrinsically disordered proteins and regions. It's estimated that at least 1/3 of all proteins have at least one unstructured region. So this dogmatic view that every protein has this one specific 3D conformation is just very confusing. Many functions also are really not the result of the three-dimensional structure, but rather something about the structural organization. And this is, I think, quite a blind spot in the structural biology community. This is where the systems biologists and synthetic biologists will claim they have the key there. But there's still this division between the three-dimensional structure, the biochemical activities, and the organizational structures and the biological roles. And I also think these two communities don't really talk to each other. So again, hopefully philosophy can help bring those together and be clear about the distinctions being made there. Also, some proteins are what's called moonlighting, so they perform multiple different functions, even though they're structurally identical if you put them in a different context. Or they might just be able to conformation switch very quickly. You have multiple realizability. So there's a bunch of problems with this idea that particular protein structure, or three-dimensional structure especially, could be necessary and sufficient for some biological functions. I think it's definitely neither. They're neither necessary nor sufficient. Best we can do, again, we have to separate which notion of structure are we talking about with notion of function. And for some of this, maybe supervenience can be feasible, but certainly no necessary and sufficient conditions and this deterministic mindset. So this idea of a one-to-one mapping between sequence, structure, and function, I think, is an illusion. But instead, I mean, once we clear out what notion are we talking about, what meaningful relations can be made between these notions, we can for sure be sure that there's some meaningful empirical correlations. Some of them might be more stringent. We could reconstruct some of them maybe as supervenience relations. But the essential first step is really to look at the right level where to make these connections, I think. So at least I hope there's a tiny solution to the mess behind this structure determines function notion that's highly problematic by itself. All right. From these relations to maybe what kind of roles, especially in terms of explanation and understanding this connections between structure and function could play. And again, these are two kind of different schools, two different explanatory projects where people are trying to explain the biochemical activities or the biological roles with their respective three-dimensional or organizational structure. And this is where the systems biologists are putting emphasis on the latter, that if regular networks turn out to be crucial to explaining development and evolution, then the reductionist interpretation may be in trouble. So I also think this is an interesting wording to put reductionist to this idea that it's all about the three-dimensional level, but even on the organizational structure level, people can be so reductionistic, but at least one notion that everything has to be reduced to the level of individual molecules and their properties. This notion of reductionism is claimed here to may be in trouble. If network-based explanations are apparently ubiquitous, it is quite likely that we will often bear the explanatory weight in such explanations as the topology of the network rather than the specific entities of which it is composed. So again, we have the display of this organizational structure notion and the individual 3D structural component and the explanatory weight can be put on these. So I think really these are two different explanatory projects on these two levels and notions. But it's not really, again, a strict boundary between these two. So as Sotra Saka here claims that how topological an explanation becomes a matter of degree. The more an explanation depends on individual properties of a vertex, the closer an explanation comes to traditional reduction. The components matter more than the structure, understood here apparently as organizational structure. So the 3D structure of individual components matter more in cases here than the organizational structure, but conversely, the more an explanation is independent of individual parts of a vertex, the less reductionistic it becomes. But again, I would be, this is one notion Sarkar has often in mind when it comes to reductionism genetics, that it's all about this molecular level. But I also think there's something reductionistic about the organizational structure kind of explanations, but still, it's a different explanatory project we can look into. But I don't think they get scot-free from the smell of reductionism either. So there's a big hype about all this molecular systems biology and synthetic biology, but I think I'll move on over this. How much time do I have left to have 5
minutes?
That sounds good.
Didn't expect it,
So let's have another table just to summarize how I think about these different explanatory projects here. So if the explanandum is the biochemical activity of an individual component, then the explanans can be just the three-dimensional structure of an individual molecule, and then you can either just by correlation-based annotation, you compare a similar-looking structure to what is in your database. You can reasonably predict some biochemical properties of this component. You can use deep learning for that, but it's not really informed based on the reductionist stream that we would have insight into the physical and chemical principles that would allow us to attribute functional features to proteins. I don't see that happening in practice at all. I think it's really mostly correlation based on, like, we have experimentally determined that this a protein that looks like this binds that component. Now with a similar looking one in our database, we'll just tag the same functional annotation feature on there. That's how I think this is being done. And there is no insight into any physical chemical principles whatsoever. When we come to this organizational structure notion of function, which is being used to explain biological roles, notion of function here I think people are either really just with riding the new mechanists wave, constructing mechanistic explanations, or which is also very trendy now, this non-causal topological explanations, where it's really the mathematical features of a network that can explain certain behaviors, tendencies, functional capacities of a network. And again, the selected effect notion of function is on an entirely different page, where natural selection will be used in the explanatory story about why certain structures have been selected for certain functions and about how this has changed. So none of this is really living up to the expectations of this classical genetic reductionism, that once we have figured out the genetic sequence, we can just basically calculate our way all the way up to the evolutionary level and skip everything in between. It even falters, I think, at this very basic level, that we don't really have any insight into the physical chemical principles allowing us to annotate functions to structures on the first level, let alone on the second one. And I also don't think that this second level here is getting off the hook of reductionism, because again, it is trying to link as a kind of necessary and sufficient conditions here, this network topology has these features. Why isn't that the same reductionist story as well? It's not reductionist in bringing it to the level of individual molecules, yes, but still to something else. So I think in a way this is also still reductionist thinking in new clothing. Godfrey Smith again has put it quite nicely, that both kinds of functions should be recognized, each associated with a different explanatory project. And that I think sums it up quite nicely. So here are some of so this is before AlphaFold, when predictive success was just limited to very small segments of molecules. Now we can do that with even like really large ensembles of proteins. And the Like here you have in red the experimentally determined structures, in blue the predicted ones. They seem to be matching quite well, a few deviations here and there. And now with AlphaFold we have that on a much larger scale with many proteins. But also sometimes it goes weirdly wrong, especially like transmembrane proteins, intrinsically unstructured proteins. It's still messy with the AI, of course. Yeah, let's not ride the wave of new mechanisms. In a way, I think this explanatory project of the organizational structure giving rise to a biological role notion of function, it kind of fits the new mechanists notion of a mechanism also because they use the term structure and function also quite prominently. Of course, as you will probably be on board with me saying that there is a lot of problems with their account of explanation, but still for a specific instance of this kind of explanatory project, I think it maps quite nicely what they claim about the entities and activities and how they bring about a certain phenomenon. I think this kind of covers what the organizational network structure people are trying to do when they explain biological things. So maybe just also skipping an example, but still, this is nothing new to draw this kind of biological network models. I mean, you can look into your favorite pathway, like here mTOR, it often looks like this, with arrows drawing between all kinds of entities, often question marks indicating there is a lot of incompleteness, and it's a useful tool, but how would you say that this kind of structure determines in a reductionist mindset any functional features? I find that hard to swallow, but still it's informative to say, oh, we know about this individual activity, we know about the dynamics of this network. There can be these different explanatory projects well on display, but also it can falter. If you have time, I can highly recommend this paper, Can a Biologist Fix a Radio, where this idea is put nicely to ridicule how a biologist might reconstruct the inner workings of a radio. If you compare it, of course it's unfair, this is a man-made artifact. The other thing is still a reasonable representation of what goes on functionally inside a radio, but it's a fun paper. Do check it out if you have time. So maybe one last remark on reductionism here. Also, we shouldn't neglect that these structures are highly dynamic. They change over time, and they're not just a static interaction network. Van Regenmortel, who has worked a lot on this idea of reductionism, especially in immunology and molecular biology, argues here that one fallacy would be to assume that the function of a gene is to produce whatever the system fails to do when the gene is absent, which is exactly ridiculed here in this paper, because a biologist would just start ripping out parts and see how the radio stops working and infer something functionally from that. So yeah, this is not a meaningful way to find out functions. This can often lead into very wrong directions. To close the circle here with the central notion of structure and function here, one last quote here: although structure and function are inseparable aspects of biological organization, this does not mean that a structure logically entails a particular function, or that the structure can possess causal efficacy in bringing about certain functions. Maybe limited to some biochemical activities, but it must also include the dimensions of time, the history of its evolutionary trajectory. So biological structure needs to be thought of as dynamic rather than static property, and I think that's also a very important lesson for the systems biology people who claim they work on these structural organizations. So time and time again, there is more going on with the sequence structure-function relationship, and the dynamics should not be neglected here. Yeah, we have talked about these different modes of explanations. Michel Morange maybe nicely put it up here that we should really not pit them against each other, and the future of biology should not be in the passive coexistence of these different kinds of explanations, nor in replacing one by another with this old reductionist idea, but in the precise interweaving of these different explanatory projects. And I think that's a very harmonizing conclusion with the different notions of structure and function, how they are related, what explanatory roles they play. And I think this is very useful, conceptually interesting, and hopefully also useful for the scientists working for these things. I'll stop here to have time for some discussion. Thank you.
Thank you. Okay, I think we can have approximately 2, maybe 3 questions. Yes. Enough?
Okay, thank you for a very interesting talk. Like in the diagram where you showcased all of the the relations basically between your terms, do you think, like you keep going from only from structure to function, but is there an explanatory role for function, like in the opposite direction, right? I think that's the key, like non-reductionistic part, because otherwise you're still going just from structure to function. Even if you say, okay, it's dynamic, you're still going only in one direction, right? But do you think you have to take into account that function by itself constrains structure?
Exactly, very good question. Yeah, so I think this bottom-up thinking is exactly this, driven by this old reductionist idea of this linear chain of determination. And actually, it would be better to not just reconstruct and go following their bottom-up approach, but exactly, again, the keyword constraints, I think, is where, if we want to think about like what kind of structures would be able to bring about this biochemical activity, like drawing this arrow the other way around, even for the biological role as well. This is very interesting to investigate, to look into how the relation could exactly the other way. And I think what we will have here is constraints that we can then explain maybe with the selected effect notion of function. So maybe I was too dismissive too quickly in saying following the bottom-up way, that it doesn't have so much explanatory weight there, but for explaining exactly why, for looking the other direction, we find what we find, these constraints could actually be explained by this selected effect, and constraints that there's only a few protein folds capable of doing certain biochemical things, and exactly there, I think constraint-based will be the route to go. So yeah, I agree. Thank you so much.
So kind of, I've also been like during the talk been thinking about that type of question. So I just want to add on something. Constraints may be interesting, but also what about recursion in some sense? Because the thing I was always thinking about, okay, why not start doing some kind of circle? For example, like you moving on from DNA to RNA, mRNA, tRNA, and then you get your proteins. But then, for example, epigenetically, those proteins then in some sense tighten or refold your DNA sequence to be expressed later in a different sense. So I think that's another avenue to explore for kind of or making it very difficult for a reductive perspective, because then you get those self-referential loops in some sense. And I'm curious, what role do they play or have structurally in the function of your presentation?
Surprisingly, not surprising, but a very deep question. Yeah, I think also this linearity of this diagram is only fitting for reconstructing the linear claim we started out, but exactly it should be more of this circling backward, intertwining. Even on the DNA sequence level, it's not all about the sequence of nucleotides, but also by itself, the three-dimensional conformation there is so important. Whether you have histones attached to it, if it's accessible at all, this makes such a huge difference. And when people just think in probably also along the lines of machine metaphors and codes, and there it's just the code, like it's the physical implementation is totally uninteresting here. But on the DNA, it's not. It's really crucial information that's part of the whole dynamics of how a sequence gets transcribed and translated in the first place, and how the whole thing follows up. And there's often these feedback loops. So yeah, definitely, I think if we want to really monitor what's going on inside a cell, inside a living organism, never will we ever have this kind of linear bottom-up chain, but it's always this feedback interactions, constant renegotiation of boundaries and starting processes, ending others. And it would be interesting to try to also graphically represent that, like have a model to capture that. I think, unfortunately, most of the time we'll end up with something messy like the mTOR pathway I showed you before. But still, we can work with this probably better than have this stuck idea of this linearity that's, I think, detrimental to understanding the processes. So that's why I really like this idea of bringing this nuanced dynamics into the picture as well.
Okay. Before we go to the last question, just one comment. Also, any thoughts or ideas that you might have, and even if we don't have enough time, during the discussion, the speakers will be kindly asked to take on the central position again, and then we can have a discussion in a way where it will be centered mostly on their presentations. This is just to say that, you know, keep on thinking about these things later on, because we can discuss them later on as well. Because I have some follow-up, potentially significant questions on this. But yes, the final question. Sorry for this.
No worries. Thank you for the interesting presentation. I was just wondering, like in the whole talk, you didn't seem to talk a lot about like the functional role of the environment in all of this. You know, like you just don't have like genetic information, but you need like this whole apparatus that like decodes genetic information, produces something. So, what are like the environmental constraints or the environmental role or the context sensitivity of all these processes? Because we only talk about it in a kind of like atomistic picture of what is DNA doing, what is proteins doing, but not in what environment. Sorry, in what environment they're doing that.
Great. I think that also follows what we just discussed, and the tradition of molecular biologists completely neglecting the environment, or such thinking of the cell as a bag of enzymes, and you basically have everything you need floating around. But actually, I also think that the environment is crucial, both like within a cell, but also from outside, that you'll have signals, structures, components, or also functional roles where it's never just this bottom-up thinking that you have all the components coded in the DNA, but it's really often external factors, components, structures, and functions coming from the internal or external environment that are parts of these interaction networks and are crucial in regulating these processes, in bringing about some of these processes in the first place. So I think this is another reductionist oversight to just start bottom up linearly and not allow for this for the environment, which rightly so is a crucial part of certainly for the biological role notion of function, maybe even on the biochemical activity. So often there it will be some external component you need to bring, like a metal or other factors. So definitely also a role for the environment. It's a blind spot in the paradigmatic molecular biology textbook picture.
Is there a generic assumption about what the environment is, or is that just...
That is the assumption, I guess.
Yeah, bag of enzyme providing everything assumption, I would say. Yeah.
Okay, thank you very much.
Thanks so much.